So, good evening, everybody. This is Gianluigi Condorelli from Italy. I'm chairing this session. There is also Yiming Wang and Yang Wing Li. Yiming Wang from the University of Singapore, Yang Wing Li from China, from Beijing. And it is really an honor to host Professor Stefanie Dimmeler for the second, as a second speaker of this series of um, seminars. And just to remind briefly that this seminar is called the High Discovery, and it is sponsored. It's organized mostly from the University of Shanghai, and the last meeting that we had was the first inaugural lecture was from Professor Eugene Braunwald, and this lecture was attended by more than 11,500 participants from 28 different countries. So it reflects the broad international platform reach and academic impact of this lecture. So here we will have approximately 45 minutes talk by Professor Stefanie Dimmeler. I will give just a few words about her CV. We all know Stefanie Dimmeler. Stefanie is ranked as one of the most cited and scientists in the world in the area of cardiovascular medicine. She received many, not only international grants, but also recognitions of her work, many prizes. She was and she has been associate or editor of many international journals in the area of cardiovascular medicine and in general on molecular medicine, and in particular, for example, European Heart Journal and EMBO Molecular Medicine, and as well as many other journals. So she is really a leader in the field of cardiovascular medicine, in particular on vascular biology and the consequences on ischemic, in particular heart disease, but not only. So she is an extremely brilliant scientist, and it is really an honor to host her in this series. So I will stop here and give the stage to Professor Dimmeler for her talk.
So maybe Gianluigi, before thank you so
Sure,
much for the
sure.
introduction. Before we start the seminar, I just want to maybe correct it that the host institute is actually Zhejiang University Second Affiliate Hospital. We
Sorry,
actually
sorry,
have
sorry, sorry.
yeah, no problem. We actually have the vice president, Professor Xinyang Hu here. Maybe Professor Hu, can you also provide us a welcome remark?
Okay. Thank you so much, Stephanie, to give the great lecture for us. And also thank you for Gianluigi to organize this, to moderate this session. And thank you, Yibing, to coordinate this great session. It's really our honor to have all of you here to have this event. Okay, thank you.
Great, thank you. Yeah, Professor Dimmeler, the podium's yours. Looking forward to it.
Yeah.
Thank you so much, and thanks to the organizers of this webinar, and also to Gianluigi for the kind words. And it's now our pleasure to introduce some novel work which we recently have done on aging. And you may ask why we are focusing on aging. So the risk factors for cardiovascular disease are quite well targeted. For example, we can treat hypercholesteremia, we can treat hypertension, we can even now reduce overweight by GLP agonists. But aging remains one of the so far unmodifiable risk factor. And you may know that about 50% of cardiovascular diseases are still accounted by unmodifiable risk factors, which are not addressed or treatable by the current medical approaches. So we wonder what is aging doing and try to more biologically understand the impact of age on the cardiovascular system. And this is particularly important because age leads to the increase of heart failure. Of course, it increases heart failure because age is inducing also atherosclerosis, leading to an increased incidence of myocardial infarction. But particularly, aging also leads to a different type of heart failure called heart failure with preserved ejection fraction, which is not, patients with this syndrome do not suffer from systolic pump function, but rather have a problem in the stiffness of their ventricle and can thereby not pump enough blood to the body. This type of heart failure is also, of course, enhanced by metabolic syndrome, but also age alone can lead to diastolic dysfunction, fibrosis, microvascular dysfunction, and vascular and cardiomyocyte stiffness. As said, this is more prevalent. This type of non-metabolic induced diastolic dysfunction is typical also for Asian and also European countries, while the metabolic induced HFpEF is more typical in the US. And we would like to understand the mechanisms underlying age-associated dysfunction of the myocardium. There are many drivers of aging. You can see here that age is induced by many processes, including telomere attrition, epigenetic alteration, disabled macroautophagy, mitochondrial dysfunction, autophagy in general. But I would like to focus today on one aspect, which is cellular senescence, which is one contributor to aging. Senescence is characterized by the irreversible arrest of cell proliferation. This is maybe a defense mechanism against the proliferation of dysfunctional cells. And actually, senescence can have a very protective role in acute injury responses. So if you have, for example, myocardial infarction, there is an induction of senescence, and this can prevent excessive proliferation of fibroblasts. And also, some factors released by senescent cells contribute to the repair. Likewise, elimination of senescent endothelial cells, for example, can aggravate liver failure induced by an injury. So you may understand that this is very important in acute situations, but if senescence occurs like in aging chronically, this can lead to a dysfunction of the tissue, particularly because these senescent cells release so-called senescence-associated secretome factors, so-called SASPs, and they can have a negative influence on the surrounding tissue and impair the microenvironment and cellular function. So we were wondering when trying to study aging, how age affects the different cells, and we used an integrated approach. We were not only looking on the cardiomyocytes, which are the lead cells leading to the contraction and the diastolic dysfunction, but also fibroblasts, the vasculature, and the inflammatory cells play a particularly important role in the age-associated induction of dysfunction of the myocardium. And actually, by Julian Wagner's approach, he is a wet lab scientist and now a junior research group leader, and David Rodriguez Morales, he is a bioinformatician. We aim to address how the cellular cells in the heart are changing during aging. And actually, I would like to highlight that aging is maybe not a one-step trajectory, but rather not a one-step process, but rather a trajectory, meaning that over age, we undergo several processes of aging, and we would like to understand this trajectory better. And currently, with the first data I will present, please note that we focused on these quite early stages of aging. This is about 18-month-old mice and about 55 to 60-year-old humans. So one needs to be taking into account that what we see are maybe the first signs of aging, and we were looking into the heart to understand what's going on. To assess which regions of the heart and which cells of the heart are particularly sensitive at this time point to aging, we developed the so-called senescence score by using established senescence scores, but also using DEGs from young, these are differentially expressed genes from young versus old tissues, and used the spatial transcriptomic data sets to identify the hotspots of senescence. you know that spatial transcriptomics is a new technology to identify the spatial resolution of transcripts across the hearts. This is a mouse heart here. And you can calculate by integrating the spatial data with single nuclear RNA sequencing data where the senescent cells are localized. You can see we identified so-called hotspots of senescence, which have a high expression of these senescence genes. And even in young hearts, you see some regions which have some senescence hotspots. In old hearts, these senescence hotspots get aggravated. You see more distribution of the senescence hotspots in the old hearts. But the question now was, what niches, so what local cells are affected by aging? And with spatial transcriptomics, you can identify individual niches. And you see here the different niches which are computed in young and old hearts. And in red, you see the niches which have a high senescence hotspots. In blue and are the niches which have a low senescence hotspot. You can appreciate that even in young hearts, we see some senescent regions, but in old hearts, particularly niche 7, showed a strong enrichment of the senescence core. And of course, the question was now, what is this niche 7? So what are the cells and the gene expression patterns contributing to these high hotspots of senescence? If you look on the right-hand side, you see here the cellular contribution. You see that the niche 7 stands out in comparison to the other niches by a high contribution. In red is always the high contribution, arterial endothelial cells. You see high accumulation of macrophages, high accumulation of smooth muscle cells, and high accumulation of active fibroblasts. Interestingly, pericytes and cardiomyocytes are reduced in these niches, suggesting that these senescent niches are particularly resembling the macrovasculature. So they are devoid of cardiomyocytes. Cardiomyocytes age later at 22 to 24 months. It's not shown here, but at 18 months, we see preferentially the senescence hotspots around these vascular niches. To see whether this prediction by this in silico data is true, we assessed the senescent cells by acidic beta-galactosidase staining. And you can appreciate these are 3 months old mice, these are 18 months old mice, that the blue color indicative of senescent cells is particularly around the larger vessels. This is a second staining, also detecting senescent cells in red here, so-called cell event. And you again see here cell event positive cells around the vessels, which are also enriched with fibroblasts. Quantification shows that these senescent cells at 18 months is particularly high in these adventitial regions and not yet occurring in interstitial regions. So I mentioned that also macrophages are enriched in these niches. And indeed, if we performed immunostainings, we can appreciate also that in these adventitial niches at 18 months, there is an increase in CD68 positive macrophages. You may know that there are 2 types of macrophages in the heart. One are tissue-resident macrophages. These are more reparative macrophages, also important for conductance, and they are important for maintaining tissue homeostasis. And bone marrow-derived macrophages, which are more inflammatory. To dissect which of these cells are more prevalent in these regions, we assessed the staining of the markers for these cells. And here you see the tissue-resident macrophages. And we made a surprising observation, namely that despite the increase of the overall macrophages, these reparative tissue-resident macrophages decline. In contrast, all of the macrophages which were enriched were bone marrow-derived, as you can see here, and they contribute to the overall increase in this macrophage burden in these vascular regions. Finally, we also assessed the gene expression in these regions, and what stands out was a high enrichment of so-called do not eat me signals. These are very interesting signals which are expressed also by senescent cells, but also we see them expressed in the old heart by many cell types actually. They prevent macrophages from cleaning tissues if the macrophages have the ligand here SIRP-alpha and binds to the CD47, then this prevents that macrophages eat the senescent cells, for example. So the high expression of these two proteins or genes would mean that we have an enrichment of cells which otherwise would be cleaned of the tissue, and this may lead to the long-term, let's say, aggravation and accumulation of these cells. Also, these macrophages had a very high chemokine and vascular adhesion molecule expression, which is typical for bone marrow-derived cells, underscoring their potential contribution to inflammation around these vascular areas. So together we found, quite surprisingly to me actually, that the first hotspots of senescence which we found at early stages of aging are around the perivascular niches, and that these niches are enriched for smooth muscle cells and activated fibroblasts, bone marrow-derived macrophages, and they express high expression of these, they express high levels of do not eat me genes, which prevent the degradation and let's say engulfment of other cells in this tissue. But these regions were devoid of tissue-resident macrophages, suggesting a dysbalance of our macrophage pool in the heart also. And I will not go into detail, but we found similar findings in very preliminary human data. Also here in these older heart samples, we see higher senescence hotspots. And again, these senescence hotspots were in the larger vascular niches, suggesting that this is a conserved phenomenon in humans as well as in mice. Together, these data suggest that the vasculature, which of course is very important in the heart, but also spans all across our body, is one of the first hotspots of aging, particularly enriched for senescence in the heart. Interestingly, in a very interesting Cell paper, which was recently published at the same time when our paper came out by Liu's group, they also found in proteomic data sets in humans that the first tissue which ages is also blood vessels, which age around age 50. So this is quite early time again. So this would suggest that the vasculature is maybe a sensitive tissue for aging. And if you consider that the vasculature spans 2 times the world of one human, and if you consider that if your endothelial cells are plated from humans, you can really fill a full soccer field. Of course, changing senescence and changing the function and the maybe secretome of these vasculatures and vascular cells may have a huge impact on body, possibly, although we haven't looked into this, possibly beyond the heart, but maybe also in other organs. So with these data in mind, of course, it's interesting to further understand the molecular signatures and changes in the vascular cells. And we were particularly focusing in our studies early on endothelial cells. In the heart, endothelial cells have very important functions. Of course, they provide oxygen and nutrients. This is since long known. But the vasculature also is important for waste export. So this is something which is so far underlooked, but it has maybe a very important function as well. Also, the vasculature can crosstalk to fibroblasts and change. They can, of course, affect the extracellular matrix, and this may be a vicious crosstalk in aging. Also, endothelial cells and the vasculature are the gatekeepers to prevent immune excess, but also allow reparative inflammation. And of course, they are very important for the regulation of vessel stability and edema formation as well. So with all of this in mind, we asked the question, what is the impact of aging on endothelial cells in our mouse hearts? So again, comparing 2 weeks and 18 months mice and selecting the endothelial cells to study the gene expression profile. And in addition to finding things which I just have shown you, which we have been also in part knowing, we made a very surprising observation, which I would like to share with you. Namely, that one of the top Go terms significantly enriched in endothelial cells in the aging heart were axon injury and neuron death. This was very surprising because you haven't seen nerves in my previous slide, and we didn't consider nerves being part in the aging process. But it's since long known that nerves and vessels align with each other, and also in the heart you see here by these immunostainings, in green the nerves, and in pink the smooth muscle actin positive vessels, in red the capillaries. And you can appreciate nerves aligning closely to these vessel structures here in the heart. So we question whether aging may have an impact of this nerve endothelial cell crosstalks. And indeed, if we simply measure axon density in the left ventricle, you can appreciate a strong decline of axons at 16 and 18 months, and a further decline subsequently at 22 months. An example you can see here on the right-hand side in young mice, these are the axons stained by 2G1. You have to keep in mind that the nerve bodies are outside, and these are the projections only into the heart. And in old hearts, you see here much less of these 2G1 staining, suggesting that this is particularly left ventricle here. Aging leads to a decline of nerve density. I do not have time to share you all of these data, but it's published, and you can find in the publication that aging reduces sympathetic, parasympathetic, but also sensory nerves, so suggesting a general anti-repulsive or neurotoxic effect in the aging heart towards all different types of nerves in mice and in humans. What is the mechanism underlying this? So we ask how aging may lead to this dysregulation of nerve density, and we found actually two mechanisms by which aging affects the nerves. One was that aging induces so-called repulsive signals, and one of the repulsive signals we found were SEMA3A. Semaphorins are important mediators of nerve repulsion. And you can see here semaphorin 3A was induced in old heart endothelial cells compared to young heart endothelial cells. If you add semaphorin 3A just to cardiac organoids, you can see that this leads to a decline in nerve density, again using 2G1 to stain the nerves. Also, if we specifically overexpress semaphorin 3A in endothelial cells, this leads in mice to a reduction of these nerves in the mouse heart, suggesting that semaphorin 3A in endothelial cells can really lead to a reduction of nerve density in the aging mouse heart. Second, we found that there is a reduction in neuroprotective signals, and what factor stands out was VEGF-B. VEGF-B declined in old heart endothelial cells, and you can appreciate if we overexpress VEGF-B in old hearts by using AAVs together with Kari Alitaro's group, we found an increased innervation in the old heart, suggesting that we can rescue the reduced innervation partly also by providing neuroprotective signals to the heart. So with all of this, we conclude that we should add denervation as one of the hallmarks of aging. You see here the typical hallmarks which I have alluded to before: fibrosis, inflammation, mitochondrial dysfunction, also apoptosis, and we think that denervation together with this inflammation around the vascular niches may play a very important role here as well. With this, of course, the question is, how does senescence play a role in it? I mentioned before that senescence and the senescence-associated secretome can play a very important role to change the local environment. And interestingly, we found semaphorin 3A, the repulsive signal, which can lead to the denervation, being enriched in endothelial cells, which we cultured chronically, suggesting that semaphorin 3A is part of the senescence-associated secretomes. And we asked the question whether senescence, which is also around these vascular niches where we see the decrease in nerve density, may contribute to denervation. To do so, to address this question, we use first senolytics. So senolytics are drugs initially discovered by Kirkland's group, and they are known to kill senescent cells and thereby can clean senescent cells from the tissue by inducing apoptosis. There are many approaches how to achieve the deaths of senolytic cells. And we use the classical senolytics, dasatinib and quercetin, because also it has been used in a phase 1 feasibility trial in a very few patients, and maybe an approach which might be at least also translationally interesting. We did so by testing senolytics in aged mice and compared, as said, dasatinib and quercetin with placebo controls in old mice hearts. And you can appreciate here that consistent with what has been published, if you treat 18-month-old mice for 2 months with these senolytics, these are the senescent cells in the old heart, that these senescent cells are really mainly cleaned. You see here a strong reduction of senescent cells. And indeed, senescence appears to be involved in the denervation, because we also find a regrowth and a reinnervation of the heart if we treat the old hearts with senolytics, the old mice with senolytics. So the reduction of senescence was accompanied by a regrowth of the nerves. What could be the biological implications? Of course, nerves have important function in the heart, and we are still trying to get more direct evidence for nerve functions, but what you can easily measure is heart rate variability. And heart rate variability reflects the response of the heart to stress. And in elderlies, it's well known that the heart variability is reduced and that this is leading to a poor prognosis. So we ask whether heart variability is changed by senolytic treatment. And indeed, we can find that heart variability is increased, so restored to the levels of the young, almost to the levels of the young, if we treat the mice with senolytics. Interestingly, we could also restore the day-night cycle, which is typical for the heart variability. You see here in young hearts, the heart variability day-night. And in old hearts, this is entirely suppressed, so we have not enough, very little heart variability on days. If we treat the mice with senolytics, we could restore this day-night cycle activity, suggesting there's really a biological response to senolytics. A second feature, of course, as you know, nerves are very important for controlling electrical stability, and it's also known that denervation can lead to silent ischemia and lethal arrhythmias. If we used the hearts and stressed them to induce arrhythmias, you can appreciate an induction of arrhythmias in the old mice with placebo treatment, but this was significantly reduced if we had treated the mice with senolytics, suggesting that we also reduce the risks of arrhythmias in these old mice. Finally, we also determined what could be the consequence on heart function. I mentioned that diastolic dysfunction, so the stiffness of the ventricle, is one of the important features of the aging heart. And indeed, if we treat the mice with dasatinib and quercetin with the senolytics, we could reduce the age-induced diastolic dysfunction. Likewise, we could see a decline in fibrosis, clearly suggesting that senolytics have a beneficial effect on cardiac aging. With this, I would like to conclude that we have found in our studies that during aging, there is a regression of axons, which is mediated by semaphorin 3A and also by the reduction of neurotrophic growth factors. At the same time, we find an increase in bone marrow-derived macrophages, which may lead together with these neurorepulsive signals to the dysregulation of the vascular niche. If we add senolytics, we can restore these vascular niches, and also, I didn't have time to show you, restore also the macrophage dysregulation, which we have seen, suggesting that this may be an approach to prevent or at least to reduce the subsequent aging of mice hearts and to improve the vascular niches. I just mentioned also that the macrophages have so-called do not eat me signals, and this may lead to the accumulation of senescent cells. Of course, we can clean the senescent cells with the senolytics, but still this may lead to the further accumulation of senescence in these regions. And finally, we proposed, although we have not data, this is ongoing, that there might be a neurovascular immune crosstalk in these vascular niches, which we are currently studying. Of course, this all looks very promising, but one has to keep in mind that senolytics are challenging. First of all, we need an optimal timing for adding senolytics, because if we have no senolytic cells, of course, then senescent cells, then this doesn't help. If we have a very high number of senescent cells, this may be detrimental, because you can, of course, understand if you have a lot of senescent cells, the killing of all of them may induce tissue damage and be detrimental. Second, we need a time point where we still have some repair capacity. So if we use senolytics, we may need to treat mice or patients, hopefully at a quite earlier time of aging and not at very late stages. So we are currently developing, and this is ongoing work, biomarker-guided therapeutic strategies to really time better the maybe use of senolytics. And we do now mouse study to see when, to what extent, with what burden of senescence we still can use senolytics with beneficial effect. Second, of course, it may be considerable that we may look for alternative strategies instead of killing the senescent cells, interfere with the cellular disorientation or dysregulation during aging by other means. And here I would like to share one project which is ongoing, which is addressing the control of endothelial cell function by epigenetics, because we were wondering how epigenetic regulation may chronically affect endothelial cells and thereby may lead to a maybe irreversible chronic dysfunction of the vasculature. And we did, for assessing this, an attack sequencing study where you can assess open and closed chromatin structures. And we found that one gene called ZPTB16 was highly repressed in aging. You can see here it was among the highest repressed genes in aging. This means that the chromatin is closed for this gene, and this gene is thereby downregulated. You see here that the downregulation is indeed true. You see here reduction in endothelial cells, expression in human and in mouse, and also at protein level. And you can appreciate if we take heterozygote mice for this gene ZPTB16, which have only a half maximal reduction in ZPTB16 mRNA expression, we still already observe diastolic dysfunction, LV mass increases, no systolic function, so typical this diastolic feature, increased fibrosis, increased hypertrophy, and also increased senescence, again, particularly around these vascular niches, suggesting a powerful impact if you reduce this transcription factor. This was confirmed by endothelial cell-specific knockouts. This is just one example. More can be read in this paper, which was recently published. You see here endothelial cell-specific knockout of ZPTB16 also induces diastolic dysfunction without aging. So these were young mice, suggesting that it has a very important effect. We asked the question, what is the ZPTB16 transcription factor doing? And found that these are the genes increased by ZPTB16 silencing. You see here many inflammatory mediators, paracrine active factors, inflammatory response, MET activation, suggesting that maybe we have effects in a paracrine manner by endothelial cells. And we tested this by silencing ZPTB16 in endothelial cells, used the conditioned media and added this to fibroblasts. And you can appreciate here that collagen expression was increased in fibroblasts which were treated with this conditioned media. Also smooth muscle actin was increased. And we could see a similar finding if we used cardiac organoids. Also in cardiac organoids, collagen and smooth muscle actin expression was induced, suggesting that the reduction of ZPTB16 really has a paracrine effect on fibroblasts in this paracrine assays. We also co-cultured cardiomyocytes and indeed could find that silencing ZPTB16 in endothelial cells leads to an increase in size of the cardiomyocytes. You see also that the endothelial cells don't look as healthy. The beating frequency, which is a typical stress response of cardiomyocytes, was increased also, suggesting that also the crosstalk of endothelial cells to cardiomyocytes is impaired under this context. Together, this suggests that ZPTB16 plays a critical role in endothelial cell function. It can promote also endothelial cell senescence if it's depleted. It's a critical regulator of fibroblast activation, cardiomyocyte hypertrophy, and finally, also we found a reduction in neural sprouting, again, consistent with this view that senescence drives also denervation of the heart. To sum up, of course, this would lead the opportunity for a therapeutic approach. And to do so, we aim to overexpress this ZPTB16 specifically in endothelial cells. And we did so in collaboration with a team with Christian Kuppert and Manfred Ogris and Oliver Müller, using a specific technology enhancing the delivery of vectors to endothelial cells. and using endothelial cell-specific promoters. And you can appreciate that ZBTB16 is overexpressed in endothelial cells fractions, and we can really improve diastolic dysfunction by this means, and we could also rescue to some extent fibrosis. So you will prevent, these are 18 months of mice, which were used to treat with these vectors, and then at 20 months, this is the fibrosis level, and this is preventing the further increase in fibrosis to the levels of the 18 months old mice. This suggests that aging is really detrimental process in aging. In the context of chronic aging, particularly senescence in the vascular niches, I have shown you that epigenetic control of gene expression, which are repressant of ZBTB16, can contribute to impairing particularly the vasculature. This is contributed to a paracrine activity and the control of fibroblasts and cardiomyocyte function, which is indirectly regulated by the vascular niche in the aging heart. I thought Gianluigi allowed me a few more minutes, 45 minutes, if I may. Otherwise, I can stop here. But if I have a few more minutes, then I would like, with the last slides, just broaden the scope, because we believe that aging is not only affect the heart, but it is systemically affecting all, of course, our body. And one aspect, I think, which is particularly important, considering that we see all the bone marrow-derived macrophages and the senescence hotspots, is that the contribution of the bone marrow-derived cells may be very important. Yeah, and you may know that particularly the bone marrow is affected by aging. All of our tissue are affected by somatic mutations, particularly the bone marrow is affected because the accumulation of mutations and chromosomal aberrations can have a detrimental function. So it's known that mutation in driver genes, these are epigenetic regulators, but also signaling molecules, can lead to the clonal expansion of the mutant cells in aging, so-called CHIP. Also, and this is the most prevalent chromosomal alteration seen, is the loss of the Y chromosome. So males lose their Y chromosomes in the hematopoietic stem cells. This happens in about 40% of males above 70 years old, suggesting that this is a very common situation. And this may, of course, affect the bone marrow cells invading to the heart and bring this into the context of the vascular lesion and may aggravate aging of the heart. So just to give you a brief background on this, so these are the CHIP driver genes. You see here the critical age-dependent accumulation of these CHIP driver genes. Also loss of Y, loss of the Y chromosome is a clear age-related process, typically seen above, as I said, 70-year-old. And I must say this is not only affecting the heart, but we and others have shown that these mutations also lead to aggravation of many cardiovascular diseases, including also atherosclerosis, aortic valve stenosis, but also heart failure. These are just is one example of many studies also from other groups to give you a glimpse on how this looks like. These are now CHIP mutations in TET2 and then DNMT3A. These are the two driver genes which are mainly affecting patients, and they are detected in about 20 to 30% of heart failure patients. So you see here in heart failure, these CHIP mutations are even further aggravated, and they have an impact on the prognosis. You see here, if you have these mutations in your blood cells, then the prognosis of these patients is much worse compared to the no mutant cells, the patients who do not carry these mutations. Second, you may say this is just a biomarker. At least animal experiments, but for many groups have shown that if you mimic this context in mice models, the reduction of these genes here, which are typical CHIP driver mutations, can lead to atherosclerosis, heart failure, diastolic dysfunction, and age-associated typical features of the heart. This is particularly shown by Ken Ward's group. We asked the question, what happened in these patients which have these mutations, and used single-cell sequencing to address the gene expression profile in these CHIP carriers. You see here DNMT3A and TET2, and in the middle the no CHIP carriers. And you can appreciate that cytokines such as interleukin-6 receptor, interleukin-1, macrophage activation markers, and the NLRP3 is increased in expression, and overall, these monocytes in these patients show a very high aggravation of inflammatory signatures. In addition, we made by using additionally in silico approaches to predict the interaction of these cells with the heart, which is published in this paper by Mariana. We also found out that some of the mutations, particularly DNMT3A, can also aggravate fibrosis. So if you block this DNMT3A expression in monocytes and then add the supernatants to fibroblasts, you see induction of fibroblast activation. And also in mice, if you use this humanized DNMT3A chip model, you can increase the fibrosis, particularly in the remote zone. And even in patients, we found that T2-1 mapping, this shows fibrosis is aggravated in patients carrying these chip mutations compared to no chip carriers. All of this is suggesting, and I must say this is now a bit speculative because we don't have the data truly in our aging models. This is ongoing studies, but it's maybe consistent with the notion that chip clearly occurs in aging and also loss of Y occurs in aging. They may aggravate the age-associated dysfunction of the heart by providing paracrine factors, which can, for example, activate cardiac fibroblasts, but also aggravate the vascular dysfunction, particularly by the bone marrow-derived macrophages, which are invading to the aging heart, which is definitely shown. So it may be speculated that this cell event positive region, that this may be further aggravated in a context where we have additionally a burden of chip or loss of Y carrying cells. I know this is speculation, but I just wanted to bring into this as a sake for the discussion. Clearly, it's shown that these chip mutation cells and the loss of Y cells have an effect on the heart, and this can lead to myocardium dysfunction and also atherosclerotic lesion formation. With this, of course, this opens the opportunity for a more selected approach. Maybe we can also use chip and loss of Y to define patients at risk for early aging and interfere, for example, with senolytics, although this has to be, of course, shown, or maybe treat more specifically with anti-inflammatory, antifibrotic substances and use a more precision medicine approach by diagnosing this chip and loss of Y alterations in our patients and in healthy volunteers, actually, because it's, of course, not only occurring in patients, but also in healthy volunteers, which are not aware of their intrinsic risk. With this, I would like to summarize and particularly thank the entire team for their studies, also the funding agencies, and I would like to highlight Julian Wagner, who really spearheaded all of these aging studies I have shown. I have no time to mention all of the people involved, but particularly David did the in silico spatial work, and we have a lot of collaborators helping us with these projects. And the clonal homeostasis team, I only had time to give you a brief glimpse on. Also, they have more interesting studies on particularly the loss of the Y chromosome head by Andreas Zeyer and Timo Speer, who is a nephrology in our department, and many people in the lab. Mariana did the study I have shown you today on the fibroblast activation. With this, I'm happy to end my presentation and thank you for your attention. I'm happy to address now open questions.
So thank you very much, Stefanie. I would just like to mention that one of the organizers is and host is Professor Xinyan Hu, who is professor of cardiology and executive president of the hospital, which is organizing these seminars. It is a second affiliated hospital of as Yibing mentioned before, of Shenyang University, and it is also the important sponsorship of the Shanghai Medical Innovation Foundation. So with this, I would like to open the discussion. If there are questions, maybe you can help us in doing this task. And of course, Xinyan, if you would like to say a few words.
Okay, thank you. Thank you, Stefanie, for the great talk. We really learn a lot. I want to ask one question. How does the innovation of the heart will affect the immune cell infiltration?
This is a very good question. We don't know yet. We have to do more studies on this. I can't tell you.
Okay, thank you.
It's a very good question because there may be the nerves also influence the macrophages. And we are currently doing time course studies also after senolytics and the regrowth of the nerves to see what is the time course. And it's very difficult to, if you just, we did one study where we just depleted the sympathetic nerves, but this is very crude, so the mice are also dying soon. And it would be better to really specifically manipulate the nerves with optogenetic methods to study this in more detail. This is just the explanation that it takes much more longer to have better answers.
Yeah, thank you. So another question is for the senolytic therapy translation. So will you do the clinical study using this therapy? If so, which kind of disease you will do first?
This is a very good question because, I think we need to understand the system a bit better before going into patients, because I said we need to have the right window. I think, of course, ideally we would treat patients, subjects which are still healthy and only have accumulation of senescent cells, and prevent them from becoming diastolic dysfunction. Yeah. But this is very difficult. So we would have to have biomarkers, and then to tell someone who is healthy to take a drug is very difficult. So maybe our idea is to use biomarkers to select for early on diastolic dysfunction patients, which have signs of senescence with SASPs in the blood, and then target senescence quite early on. I think if you come too late, it will be not a good way. In mice, we now study senolytic treatment at 22 and 24 months, and then look on the detrimental response in tissue to know exactly how many senescent cells burden and SASP we can tolerate to have a negative effect. Because I'm worried if we have heart failure plus aging, we may have a too high burden of senescent cells.
Okay, thank you.
Ibin.
Thank you, Professor Dimmeler. This is an amazing presentation, and I just heard it recently. I still feel so fascinated by many of the findings. I particularly was struck by your observation of this cell, sort of a senescence hotspot in even the young heart, right? So you can detect them even though it's a low frequency, but you see them based on the senescence score. So I wonder, what is the trigger of those senescent cells in the early young heart, and what is the risk factor that promote this expansion of the senescence hot spot or hot niche as we go through the aging process?
It's difficult. I can only speculate. Let's say that the finding that these hotspots are always around the larger vessels suggests that must be some stress at the vasculature. Of course, the vascula is exposed to stress, like by each blood in pumping, or you have a lot of stretch on the vasculature. It could be that this is with piezos or something like this, then promoting the sensitivity to senescence. It must also be that there are some cells being maybe more sensitive to senescence than others, and the smooth muscle cells seem to be quite sensitive. If we look in detail, these are the smooth muscle cells, which are predominantly senescence early on. It had been shown that some synthetic smooth muscle cells have more senescence, so it could be that it's a switch in phenotypes which promote senescence. But I still don't know what is the initiator of all of this. This would just explain why we have a higher sensitivity there, and would not explain why, what is the stressor. I don't know. Also in this Cell paper, they don't do only speculate why the vasculature is aging early, and they have no mechanism behind this.
Yeah, so related to this question is also, as you indicated earlier, the aging process is not linear. It's actually, there's this sort of a bump right? There's a accelerated aging for instance, in human beyond 60, and then there's another kind of a slope of aging. Can you speculate that you see similar sort of a different rates of aging in different cohorts, early or versus late, in the animal models?
So we don't have, we are currently preparing single-cell sequencing data across the whole age span. But what we have is histology data. And there are different, I would say, tipping points in aging. The first comes at around 18 months, where we see denervation, this senescence in the vascular niches. Then at later stages, 22 to 24 months, we see this senescence then of cardiomyocytes. So we believe that this is a later stage phenomenon, while these vascular aging phenomena are earlier. And this is the same actually in humans. So we have some preliminary data in human sections, which of course are more heterogeneous, but there is the same trend that early on cardiomyocytes are, funny enough, not so much affected. They are later affected, suggesting that we have very different responses. And it would be interesting to understand better why this is the case. So maybe the hypertension, which is induced then by the vascular senescence and the vascular injury, then drives the cardiomyocyte failure, could be a hypothesis. We don't have the data yet.
That's fascinating. That means that you know the even though the speed or slope is different, but also the composition or the characteristics are perhaps different in early stage versus late stage aging. That's fascinating. And then just the last question is, we know some of the risk factors contributing to the common shared cardiovascular disease, like for instance, the metabolic disorder and chronic hypertension and so forth. Would those risk factor or external perturbation can accelerate the senescence niche expansion and progression?
So it's studied for diabetes. So certainly the diabetes can lead to denervation of the heart. This is known. It's also known that diabetes can accelerate senescence. I'm not aware that somebody looked in detail in the senescence hotspots we described, but I would expect that this is also affected in diabetes. We have some studies in just if you add high-fat diets, I don't think that it's doing the same phenotype. It's not it's different than the aging phenomenon we have seen. So with the Scatarella mouse model, we see a bit different features with nitric oxide synthase inhibition and simply adding high-fat diet. So I think there the aging aspect has additional different features, but diabetes really is sharing more similarities.
Thank you. Thank you so very much.
Thank you. Are there other questions from the audience? So maybe it's a little bit late. So
I have a question.
here we go.
Oh,
Yes,
hello.
please.
Oh, great talk, Professor Stefanie. I have because I'm very interested about the macrophages. My project is about the macrophages in heart failure. So my first question is about in your slides, you mentioned that the bone marrow derived macrophages have the high expressions of the signal like "don't eat me," but the resident macrophages, the relative lies, these resident macrophages have this expression. So why these two different type of macrophage have this different signaling? Because as we expected in the vascular niche, maybe the resident macrophages have the first phenomenon. I expect. So why this long-term, the long distance, the bone marrow derived macrophage have this signaling? So this is the first question.
Maybe I did not communicate this well. So the "do not eat me" signals is interestingly expressed on both. So it's not specific to one of the two. It's just that the ratio of the bone marrow derived and the tissue resident is different. And this is quite interesting because the tissue resident go down despite an augmentation of the overall macrophage population. But they seem to express overall these "do not eat me" signals, which maybe is induced by this environment in these niches. and therefore they are expected to have difficulties in cleaning the tissue, both populations. The problem is in single cell sequencing, these populations are quite small, and the mapping to them, so it would be nice to have a bit better resolution data to really look into the detailed macrophage populations, which we haven't yet been able to do so. But the global expression was not so different.
Oh, okay. Because I see in your humans data the heat map, I see the LYVE, the positive microphages also have the top. So I think maybe that also have this signaling, the high
Yeah,
expression.
they also have it.
Yes.
Yeah.
Yes,
Yeah.
in human data, I
Right.
see that. The second question about the chip in aging. Because researchers, they have explored the chip in the heart failure, but whether have the difference about the chip in HFpEF and HRAF, these two different heart failure, the chip.
What do you mean the transition from HFpEF to HFpEF?
Yes,
or why?
Yes, because I think the HRAF is a severe inflammatory disease, but the HFpEF maybe it's not severe like the HRAF. So the degree, the inflammatory degree is not the same. So whether the chip in the aging patients or in these two different type of disease have the different mechanisms of the phenotypes. Because most studies they only they explored the chip have the association with the aging or the heart failure, but whether these two different type of heart failure have the different association or have the different mechanism about the chip.
It's a very good question. I think the HFrEF have of course a lot of inflammatory cells, and it changes inflammation because of the acute injury and the subsequent changes, because you kill a lot of tissue-resident macrophages after acute myocardial infarction. So in the border zone, at least, these protective macrophages are depleted. At the same time, you have an invasion of the bone marrow cells. So this is a huge change. I think in these age charts, and also to some extent in the HFpEF, we have single cell sequencing from HFpEF patients. These macrophage changes also occur, but they are not, you are right, a bit different, but they are more systemically done. And also in aging, we see them around these vascular niches, which is an entire different distribution.
Yes.
So I think we have to learn more, and at least from a biological perspective, it would be very interesting for me to understand better how they differentially influence the different regional effects also. This is, of course, for therapeutic applications. You may say just go with anti-inflammatory treatments, but I think biologically we have to first understand better what's going on to get a better hint on what's the cause and consequences and the local effects.
Yeah. I'm very interested about this part. Also explored some phenomena. So want to answer to these questions, whether have these differences.
Yeah.
Thank you. Other questions from the audience? Professor Hu, would you like to say something to close the close remarks, as a close remark, and Yibing also? Professor Wong, would you like to say something to close the seminar?
I'll give this honor to Professor Hu.
Okay, thank you again for Stephanie for the fantastic talk, and also thank Gianluigi and Yibing to co-organize this session. Thank all of the audience to join us. So we will close the session here, and we welcome Stephanie and Gianluigi to visit Hangzhou in person, and looking forward to see you in Hangzhou. Thank you again.
Absolutely.
Thank you very much for the invitation.
Thank you so much.
And,
I
goodbye
see you
also,
soon.
Yibing and Gianluigi.
Absolutely.
Bye-bye,
Thank you so
Stephanie,
much.
Yibing.
Thank you.
Yeah.
Bye. Bye-bye.
Bye-bye.
Bye-bye. Thank you.
Thank you.
Ingrid,
Bye-bye.
thank you
Yeah.
for the
Thank you.
organization.
Bye-bye. Yeah. Thank you so much. Thank you. Bye-bye.